Hello everyone. First of all, I would like to thank you all for watching this video, subscribing my channel, and also sharing this, liking it, and also sharing your comments even. And if you have not done this all, please do subscribe my channel, like my video, and even do share your comments. That will be a kind of encouragement for me and also a kind of improvement for me. So quickly we will start with our video today. So in the previous video, we have seen Mendel's first law, the alternate names for that, and why is it called so, the numericals based on that, and even we have checked some board questions that were frequently repeating based on this concept. So today we will start with Mendel's second law, and Mendel's second law is also called as law of independent assortment. And now we'll see like why is it called so. Law of independent assortment. When we talk about independent is nothing but it doesn't depend on anybody. Assortment is like getting segregated or it is getting expressed on its own, separating on its own. So we all know like genes, every gene will be having one allele pair. And this allele pair, whenever it is forming a gamete, it will try to segregate itself and get independently expressed in the individual. Like for example, if you take, there is an allele pair for round and we are, let us crossing it with a wrinkled seed. Whenever it is getting expressed in an individual, these alleles get separated into gametes and then when they fuse, they come into the zygote together. Now when they come together into a zygote, never this round allele is going to blend up with the wrinkled allele and come out with a different shape. Or suppose if it is a tall height plant crossed with a dwarf plant, never the tall height is going to blend up with the dwarf height and come out with a different height. So that is what we call it as alleles are independently expressing themselves. So that is what we call it as law of independent assortment. So here it says like during a dihybrid cross. So whenever we are crossing two pairs of traits, because Mendel's second law, we even call it as law of dihybrid inheritance because it involves a dihybrid cross there. So here we have that alleles are independently assorted. That is what it says. An assortment of each pair of traits is independent of the other. They don't blend with each other. Now, why is it called as law of dihybrid inheritance? We have seen like it involves dihybrid cross. So that is why it is also called dihybrid inheritance. Now we will see the diagrammatic representation of law of independent assortment, which almost explains the law there. But still, I would like to help you out in a better way by working it directly so that you can know like how to cross a gamete and how to put it in a Punnett square. So after this is done, we will quickly see into the working of Mendel's second law. So when we take the parents, let us start with the Mendel's second law. We know like every law has got two generations there, F1 generation and F2 generation. So when we start with F1 generation here, the parents selected are round and yellow and they crossed it with wrinkled and green. We can also have the other ways like in NCERT and all, they give round and green crossed with wrinkled and yellow. So whatever it may be, whenever we select such kind of parents, here the parent selected was round and yellow. So you can see the shape of the seed round and we have the color of the seed that is yellow. So that is one parent. Second parent, you can see immediately the wrinkled seed there. Shape is wrinkled and color of the seed is green there. So here two contrasting characters are selected. One is seed color, the other one is seed shape. That is why it is called as a dihybrid cross. So when parents are having two different contrasting characters and you cross them, such kind of cross, we call it as dihybrid cross. So as usual in F1 generation, we know like all dominant characters only will be expressed and all the recessive ones will be suppressed there, though the alleles are there in the plant's body. So here you get the F1 generation. All of them are round and yellow, but they are not pure round and yellow. They are heterozygous round and yellow. Like you can see, heterozygous round is capital R, small r. Heterozygous yellow is capital Y, small y. So that is how we get heterozygous round and yellow in F1 generation, which indirectly indicates that round is a dominant shape for seed shape and yellow is the dominant color for seed color. Now we have the F2 generation proceeding because Mendel is not happy with F1 generation because he has seen like only round and yellow is getting expressed. What about with the wrinkled and green? Why wrinkled shape is not expressed and why green color is not expressed? Only the one of the parents' phenotype is getting expressed. So that made him curious and again he continued with F2 generation as usual. So whenever he continued with F2 generation, we can have 4 different kinds of gametes here. Actually, we have 4 different alleles here, like capital R, that is round allele, small r, that is allele for wrinkled shape, capital Y, that is for allele for yellow, small y, allele for green. So we have 4 different alleles. So there are 4 different kinds of gamete pairs that can be possible there. Like you can see here, round can go with yellow, getting one gamete pair there. So round and yellow is one gamete. Round can even go with green. So you can see here, capital R, small y, that is second gamete. And wrinkled, similarly with wrinkled. Wrinkled can go with yellow, small r, capital Y. And wrinkled can also go with green. That is the 4th type of allele. So here we have 4 different kinds of possibilities of gametes. Round and yellow, round and green, wrinkled and yellow, wrinkled and green. Now after crossing them, we get these kinds of gametes then. So when they are crossed, we will see this crossing in detail in the working when we are doing with the working. But as of now, I'll just show you like after crossing, how many of the plants showed round and yellow and how many of them have shown round and green and how many of them have shown wrinkled and yellow and wrinkled and green. So there were 4 different possibilities of the zygotes that appeared. Some seeds showed round and yellow color. Some seeds were round and green like this. And some seeds you can see they were wrinkled but yellow in color. Some seeds were wrinkled and green in color. So when we count round and yellow seeds, you can see around 9 of them are there out of 16 zygotes that appeared there. We had 16 zygotes coming up in this Punnett square. Out of that 16 zygotes, we had 9 of them were round and yellow. 3 of them, you can see 3 of them from this were round and green, and another 3 of them were wrinkled and yellow, and only one was wrinkled and green. So here Mendel has released his phenotypic ratio, that is 9 is to 3 is to 3 is to 1, where 9 is for round and yellow, 3 is for round and green, 3 for wrinkled and yellow, and 1 for wrinkled and green. So that is how we had these kinds of possibilities, and we'll quickly see that working after this. And now before that, we will just see what are the observations for Mendel's second law. So when we talk about observations of Mendel's second law, as usual, F1 generation, what were the plants obtained? What were the progeny obtained? Progeny is nothing but the young generation or the offspring that we get. So in F1 generation, all the pea plants showed dominant characters. What were they? Round and yellow seeds. Now when he self-pollinated this F1 progeny, remember in F1 generation, parents are cross-pollinated, and in F2 generation, parents are self-pollinated. So this F1 progeny, when they were self-pollinated, the plants appeared in 9 is to 3 is to 3 is to 1 ratio, wherein he could see There were round and yellow characters in them. Some seeds were round and green in them. Some seeds were wrinkled and yellow, and some seeds were wrinkled and green. But none of them could blend with each other. As I told you earlier, they were like independently assorted. Like round is appearing as round, wrinkled is expressed as wrinkled, yellow as yellow, and green as yellow, green. But not mixed colors. We didn't get any mixed colors, nor we didn't get any odd shapes other than round and wrinkled. So that is why it is also called as independent assortment. Now we will see the working of Mendel's second law. Generation and F2 generation. In F1 generation, as I told you, we need to follow these headings: parents phenotype, parents genotype, gametes, crossing of gametes, and F1 generation. The same thing we have done even for Mendel's first law also. So the parents selected here are pure breed of round and green plants. They are cross-pollinated with pure breed of wrinkled and yellow. So how will we write its genotype? As it is pure breed, it is homozygous. So we go capital R for round and small y for green. Because green is a recessive gene and it is in homozygous nature, so we put small y, small y. Another parent we have is wrinkled and yellow. So small r because wrinkled is recessive and capital Y because yellow is dominant. Now we are done with the genotype. Now we need to come with the gametes there. So when we are forming the gametes, gamete will try to take all the possible characteristics into it. So we have here two characteristics for the first parent: round shape and green color. So when we are making the gamete, we need to form the gamete like this: round, capital R for round, and green for y, small y. Similarly, we get another pair, capital R, small y. So now here gametes are ready for the first parent, that is capital R, small y, capital R, small y. So round and green. Same case with the next parent also. So next parent, what will you write? It is wrinkled and yellow is forming one gamete, and again wrinkled and yellow is forming second gamete. So in this way, now two parents gametes are ready. Now I have already put them in the Punnett square, as you can see here. So now we need to solve them, cross them. So first parent gametes capital R small y, capital R small y. I wrote it here. Second parent's gametes, you can see small r capital Y, small r capital Y. Now we need to cross them, this with this. So when we are crossing them, generally children have a tendency to write like this. This is how some children have a tendency to write like this. So actually it should not be written like this. The alleles for one characteristic together and the alleles for another characteristic should be together. But here what is happening? We have small r capital Y, where one allele is for the shape, another allele is for the color. Immediately again shape and color, not like this. We will put both the alleles for the shape together, that is capital R, small r. As I told you, we need to start with the dominant character always. So first we will try to put the same alleles together, that is capital R, small r together. In that again capital R should come first, followed by small r because dominant it is, followed by recessive. And then now same thing we repeat it for colors. So we have capital Y, small y there. So we write capital Y, small. So this the first zygote here. The same thing will repeat even for others also because the gametes are same. That is why we will be having the same kind of zygote pair there. So I'll just write it here. We'll type there. So we have capital R, small r, capital Y, small y. The same thing here we will do. Capital R, small r, capital Y, small y. So it is nothing but, children, here I am just crossing it. The small r capital Y with this gamete. Again the small r capital I with this gamete. That's all. Similarly this one with these two. and similarly this one with these two. That's all. We need to just combine them and write it. And whenever you are writing it, see that capital letters are in the beginning followed by small letters. Both the R's are together and both the Y's are together. So when F1 generation, now we see all the plants are heterozygous, round and yellow. Heterozygous because they are not same, identical alleles are not there. Capital R, small r stands for heterozygous and it stands for round. So I wrote heterozygous round. Capital Y, small y stand for yellow color, and that too it is heterozygous yellow, not homozygous yellow, because we have alternate alleles there. And this yellow will always try to suppress the green color and it will get expressed. So as usual in F1 generation, all the dominant characters will be expressed. Now the same F1 progeny will be the parents for F2. So we have heterozygous round and yellow, self-pollinated with heterozygous round and yellow. So this is what we got in F1 generation. Capital R, small r, capital Y, small y. Similarly, capital R, small r, capital Y, small y. Now we need to form the gametes for this. Whenever we are forming the gametes for this, gametes, as I told you, they will try to take up all the possible combinations that are possible. So here we have 4 possible combinations. One is round, wrinkled, yellow, green. So this seed shape can go with 2 colors there. Like round can go with yellow, round can even go with green. Similarly, wrinkled shape can go with yellow and wrinkled shape can go with green. So we can have round and yellow seeds, we can have round and green seeds, wrinkled and yellow seeds, and wrinkled and green. So that I will just put it in the form of gametes. Capital R, capital Y, round can go with yellow, capital R, small y, round can go with green when it is forming the gamete there. Now we will take up wrinkled, what it is doing. Wrinkled can go with yellow, then wrinkled can go with green. So we got all the 4 possible gametes. The same thing we have for the second parent also here. So whenever we are writing second parent, the same thing. Capital R can go with round, can go with yellow. Similarly, round can go with green. Wrinkled can go with yellow. Similarly, wrinkled can go with green. So wrinkled yellow, wrinkled green. So these gametes only I have written it again in the Punnett square. You can see crossing. When I am crossing them, round and yellow, round and green, wrinkled and yellow, wrinkled and green. Similarly, round and yellow, round and green, wrinkled and yellow, wrinkled and green. Now when we cross them, check this, follow the cursor here, this one, this gamete, you need to cross them with all these 4. After filling first line, go to the second gamete, cross them with all these 4. So that will fill second line. Go to third gamete, cross them with all these 4. Then third line will be done. Check the small gamete, last one, cross this with this 4. You will get fourth line. That is how we need to cross them. So first one I will do it now, you can see. So it is capital R, capital R, capital Y, capital Y. How did I get this? I crossed this with this. So capital R, capital R, capital Y, capital Y. Now this with the second one. So we have here capital R, capital Y, small y. You can tell after me or you can even do it in your book also without looking into this. And after you are done, you can just check this with the video. So when you do third one, capital R, small r, capital Y, capital Y. Similarly here also, capital R, small r, capital Y, small y. Now we'll go with second line. Second line with this gametes there. Capital R, capital R, capital Y, small y. Similarly, capital R, small y. Here, capital R, capital Y, small y. Here, capital R, small y, small y. Now third line with this wrinkled one. So we'll get capital R, small r, capital Y, capital Y. I'm just crossing this third gamete with this all these four. So now here when I cross them, you can see this small r capital Y with capital R small y. So here capital R small r, capital Y small y. Third one small r, capital Y, capital Y. Here also small r, capital Y, small y. Now come to this again. Fourth gamete. Now we are doing with the line over there. So capital R, capital Y, small y. Similarly here, capital R, small y, small y. Here you see small r, capital Y, small y. Small r, small y, small y. Now we need to, after crossing this, we will check what are these gametes growing out as. How will they express themselves? What visible character are they showing? So first one you see it is round and yellow, round and yellow, round and yellow, round and yellow. Similarly here, round and yellow, round and yellow. Now next third line, round and yellow, round and yellow, round and yellow. So finally we are getting 9 round and yellow here. First line 4 we have, then 5, 6, 7, 8 and 9. So 9 round and yellow we got. Next we move on to how many round and green. First line we don't have any round and green. Second line you see 1, and 2 round and greens are there, 1 2. And third line we will see any round and green, no. Fourth line we have here one round and green, so we have 1 2 and 3 round and green we got it. Now wrinkled and yellow. When we talk about wrinkled and yellow, you can see, so we'll just see here now wrinkled and yellow. First line we don't have any wrinkled, second line no wrinkled, third line we have here wrinkled. So wrinkled in yellow, number 1 wrinkled and yellow, 2 come to third line wrinkled and yellow, 3. So 1 2 3 we got wrinkled and yellow 3. Last we have wrinkled and green 1. So finally the phenotypic ratio that is given is 9 is to 3 is to 3 is to 1. So this is how we work on Mendel's second law. Next we will see what are the numericals that will be coming up on this Mendel's second law and the recapitulation for the day. So now we will see the numerical that is based on Mendel's second law. So when we talk about this numerical, I just gave you a numerical here. You can see Mendel crossed pea plants with round and green seeds with wrinkled and yellow. And how many plants did he get? Finally, he got 320 plants in the F2 generation. So this is the problem that is given. So I have taken the same theory part and I have put it in the numerical here. So the parents selected are round and green. They are crossed with wrinkled and yellow, and finally he got 320 plants in F2 generation. Now the question is, how many pea plants showed round and yellow, and how many showed round and green, and how many showed wrinkled and yellow, and how many showed wrinkled and green. So when we see about this plants, like how many showed round and yellow, round and green, wrinkled and yellow, wrinkled and green, here they are asking you regarding the ratio. So phenotypic ratio has to be applied. So what is that phenotypic ratio that we have learned? 9 is to 3 is to 3 is to 1. So here when we look at the solution, you see total plants 320 and the phenotypic ratio 9 is to 3 is to 3 is to 1. So what did I tell you? We need to add them up all. So 9 plus 3 plus 3 plus 1, it's coming up to 16. So divide total number of plants by 16. So the x value you will get 20 there. Now that x value you will substitute here. So how is that we will know what is 9 for, what is 3 and all? We got it from already from the theory there. Round and yellow will be 9, round and green will be 3, wrinkled and yellow will be 3, wrinkled and green will be 1. If they don't give you the same kind of parents there, then how to apply this kind of phenotype? Like here, I have taken directly from the theory. Suppose if they give you tall plant with wrinkled seeds is crossed with dwarf plant with round seeds. Tall plant with wrinkled seeds is one parent. Dwarf plant with round seeds is another parent. So in this, what are the dominant characters there? Tall is dominant, round is dominant. So whenever we talk about this 9 value, 9x, both dominant characters will be expressed. Whichever kind of question they give you related to Mendel's second law, whichever numerical they give you, whatever trait they may select, blindly you can remember like this 9 will be related to both dominant characters. So both dominant here will be tall and round according to the problem that we discussed. Another 3 you see, one dominant and one recessive. So one we can take it as tall, the other one we can take it as wrinkled. So tall and wrinkled you can give it as 3. Another 3, one is recessive, the other one is dominant. So we can take there wrinkled and we can take tall. And the last one will be both for recessive characters, that is dwarf plant and wrinkled seed. That is how you can match it. So always 9 will be for both dominant characters, 3 will be for one dominant, one recessive, another 3 for recessive and dominant, and then last one for both recessive characters. So this you can generally think and apply if they give you any kind of numerical. But here we have taken round and yellow plant, 1 and wrinkled and green. So we got 9, x value as 20. We divided it total number of plants by 16. How did we get 16? We added these all, 9 plus 3 plus 3 plus 1, which is the phenotypic ratio for second law. Now this x value we are getting 20. Now we substitute this, multiply this x value. 9 into 20, 180 plants. Round and yellow were 180 out of the 320 plants. Round and green were 60, 3 into 20. Wrinkled and yellow, 3 into 20, 60. Wrinkled and green, 1 into 20, 20. So when you add this up, you can check it. 180 plus 60, 240 plus 60, 300 plus 20, 320. So this 320 is coming up there. So that is how you can check whether the problem that you have done is correct or wrong. So for Mendel's first law, you need to divide total number of plants by 4. For Mendel's second law, you will be dividing total number of plants by 16. For Mendel's first law, 2 types of ratios will be there depending upon the question. You should think that I have discussed in Mendel's first law video. If you check that video, go through that video, you will get the details like how to apply phenotypic and genotypic ratio. So that's all for the day. As I am ending the day, I have recapitulation for you. So just go through the questions that I have shared. And then if you have any doubts related to this question, you can share that in the comment box. I'll be happy to hear from you. And please do subscribe my channel and press bell icon so that you will be getting my updated videos then. Thank you, children.